Basically, plasmids are small circular pieces of DNA. They replicate independently from the host cell's chromosomal DNA. Interestingly, despite being able to self-replicate, plasmids are not considered alive. While they can be found in eukaryotes and archaea such as plants and yeast, they are mainly found in bacteria. In nature, plasmids have a gene that provides an advantage regarding the overall fitness and survival of the host cell. Specifically, this often includes an antibiotic resistance gene. Moreover, they all have an origin of replication, responsible for controlling the number of copies of the plasmid. Lastly, natural plasmids can provide virulence, resistance to antibiotics, and degradative functions. Plasmids in the lab, on the other hand, are designed to introduce foreign DNA into a cell. Artificially made plasmids in the lab, at the minimum, always have an origin of replication, cloning site, and selection marker. Due to the ease at which it is to modify plasmids, as well as the fact that plasmids are able to self-replicate within the host cell, this makes them useful and convenient tools for biomedical engineers.
A restriction enzyme is an enzyme that cleaves DNA into fragments at or near specific recognition sites within the molecule known as restriction sites. To cut DNA, all restriction enzymes make two cuts, one through each sugar phosphate backbone of the DNA double helix. Restriction enzymes are mostly present in bacterial cells or in archaea to provide defense against viruses. The restriction enzymes selectively cut up foreign DNA in a process called restriction. Meanwhile, host DNA is protected by a modified enzyme that modifies the prokaryotic DNA and blocks cleavage. Together, these two processes form the restriction modification system. They target specific sequence of DNA in the strand called restriction sites, usually 4 to 8 base pairs in length. The length of sequence will determine how often these base pairs will appear in the genome. Most of these sequences are palindromic, which means it reads the same forward and backwards. Restriction enzymes likely evolved from a common ancestor and became widespread via horizontal gene transfer.
Scientists have found many uses for manipulating plasmids to introduce new genes into organisms. The process by which scientists take a plasmid from one bacteria, add a section of DNA that they would like to test, and put it into another bacteria is known as transformation. The first step of transformation is to use restriction enzymes to cut the section of DNA that is to be tested from an organism, like a gene coding for insulin. Then, restriction enzymes cut a plasmid so that the opening leaves two sticky ends for the target gene to attach to. Sticky ends are single-stranded pieces of the plasmid that have specific base pairs that let new DNA segments attach easily. Ligase seals the ends of these bases together. Once a plasmid is together, it is introduced to another cell by one of two different processes. The first involves placing the cells in a very cold calcium chloride solution and adding the plasmids. Then, the cells undergo a heat shock, where they are heated to 42 degrees Celsius for just under a minute, which destabilizes the membrane and allows the plasmid to go through more easily. The second process, electroporation, provides the same result by subjecting the cells to an electric shock of 12,000 volts per millimeter. Now, the cells that took in plasmids have returned to normal. Scientists can distinguish between those that were transformed or not by introducing the cells to an antibiotic. If the plasmid, which held an antibiotic-resistant gene, was taken up successfully, colonies of those bacteria will occur. In this way, scientists have learned how to express genes of different organisms at a much higher rate through bacteria to make products such as insulin, human growth hormone, or other proteins.
Whenever you're ready.
Let me tell you about plasmids, circles of DNA, bacteria, have them all inside. They're self-replicating genes that move from cell to cell. They change genetic makeup Vary the gene pool. Let me tell you about plasmids.
Let me tell about plasmids.
Let me tell you about plasmids used in biotech. Transformation. Look for palindromes. Restriction enzymes cut them up. Introduce new genes. Drugs select colonies that have been transformed. Let me tell you about plasmids. Plasmids. Let me tell you about plasmids.